name is Tracy Ying. I work on the marketing team here at GenScript. We're very excited to have guest speakers from University of Miami, Karolinska Institute, Stanford University, and Hunter College of City, University of New York. We will cover topics in HIV research, liver cancer immunotherapy, extracellular viscose generation, and more. Before I hand over the mic to today's guest speaker, Dr. Anna Pacita, I have a few housekeeping items to cover about this presentation. First, today's webinar will be available on demand after the live session and is accessible on GenScript website. We will upload the slide deck as well. Next, we'd love to hear from you during today's presentation. If you have a question for our speaker, please feel free to send it through the chat box and or tweet GenScript. We'll be answering questions at the end of the session. And last, we'd like to encourage you to complete a quick survey after the webinar and share today's webinar with your social networks. Now, let me introduce Dr. Anna Pacita. Dr. Anna Pacita recently joined the Karolinska Institute in Stockholm to work as assistant professor and managing director of the pre-GMP facility in the Department of Laboratory Medicine. She received her master's degree in molecular biotechnology in 2007 from Bologna University, Italy. She then moved to the Karolinska Institute, where she received her PhD in immunology in 2012. Dr. Pacita joined the laboratory of Dr. Steven Rosenberg at the National Cancer Institute, NIH, in 2013, and her research was focused on the isolation of TCR genes from mutation-reactive tumor infiltrating lymphocytes in solid cancers. So without any further ado, I'd like to kick things off by welcoming our guest speaker, Dr. Anna Pacita. Over to you.
Okay, thank you, Tracy, for the nice introduction. So the title of this webinar is Identification of Relevant Antigenic Targets for Successful Liver Cancer Immunotherapy. I prepared an outline for this talk. So first of all, I will give some short background on hepatocellular carcinoma and how the liver microenvironment affects the immune cells. And then a few examples of promising immunotherapies for HCC patients, like cytokines, monoclonal antibodies, TILs or CARs therapies. Then I will go more in detail in our current hypothesis, where we think that successful immunotherapy depends on targeting the relevant antigens for cancer progression. And I will also describe our screening approach for hepatocellular carcinoma here at Karolinska Institute. In particular, we focus on specific genes that are clinically relevant for tumor progression, and we identify T-cell receptor able to recognize neoantigens within these genes. So hepatocellular carcinoma represents approximately 90% of all cases of primary liver cancer. The major risk factors are chronic alcoholic consumption, viral hepatitis, non-alcoholic fatty liver disease. And depending on the geographic area, these factors may be more or less prevalent. So for example, in Asian countries like China, viral hepatitis, meaning hepatitis B, hepatitis C, hepatitis D, are still very prevalent, and they are associated with the hepatocellular carcinoma. In the Western world, like in the United States, for example, other risk factors like obesity or diabetes are emerging for hepatocellular carcinoma. This type of cancer is staged in a simple way, in a sense, because the major analysis that is done is the numbers and the size of the lesions present in the liver and the overall functionality of the liver in this patient. So when the cancer is detected earlier on, so when there are few and small lesions, the best therapeutic approach is actually surgery, or ablation. And in some cases, it's possible also to have a liver transplant. Then if the cancer is identified at a later stage, then The therapies available are not that effective, is most likely going just to prolong the life of the patient for a few months, like, for example, sorafenib is a kinase inhibitors. And then if the cancer is detected at a very terminal stage, then there is only supportive care available. The problem with liver cancer is that most of the time is asymptomatic until the very last or late stage. So the patients are not identified early enough. If early detection happens, usually is in patients that are already in risk groups, so patients that are known to have chronic hepatitis or cirrhosis or other liver diseases. So recently, another study started to evaluate the presence of mutation in certain genes present on hepatocellular carcinoma and try to correlate the mutation in these genes with the pathological feature, for example. And interesting genes emerge, like, for example, TP53 and beta-catenin. So one aspect that needs to be kept in mind is that the liver itself has the capacity to induce tolerance against antigen, usually antigens that come from the gut, from the food that we ingest. And there are specific antigen-presenting cells in the liver, like Kupffer cells or Langerhans cells, that already express for example, PDL1 and PDL2, and secrete also inhibitory cytokines like IL-10 and TGF-beta. So this environment is an environment that prevents activation of effector cells. And this is a physiological aspect of the liver. But if the liver starts to develop cancer, so if there is a cancer cell that starts to express cancer antigen now, the problem is that also T cells that could recognize this antigen are actually suppressed by this inhibitory environment. So the main goal when one wants to have an immunotherapy for hepatocellular carcinoma is really tilt the balance of the liver tolerance. So try to get the immune response to be active again in this environment and to target specific antigens that are only present on cancer cells. So several immunotherapy approaches have been quite promising, actually, tried for hepatocellular carcinoma. And in these slides, I will just briefly describe the most important ones. So some is like monoclonal therapy with monoclonal antibodies that recognize antigens that are expressed on the cancer cells, like anti-glypican-3 and anti-mesothelin. Then checkpoint inhibitors. So these are also monoclonal antibodies, but they recognize, for example, PDL1 or PD1 and CTLA4. So there are molecules that don't aim at the tumor cells, but they aim at the T cells, and they try to release the blocks on these cell types. And for hepatocellular carcinoma, recent clinical trials show an objective response about 20%. So it's still a minority of patients, but it's a very promising result. And other therapies that have been tried are cytokine-induced killer cells. These are a type of NK cells. Then vaccines against tumor antigens, but also viral antigens, because the hepatocellular carcinoma is very often associated with chronic hepatitis infection. And then therapy with the modified T cells. And these cells can be modified by expressing a CAR or a T cell receptor. And in this type of therapy, I will talk a little bit more in detail. So what is adoptive cell therapy with modified T cells? So we can have, like it shows in this slide, different type of it. And the difference is really like how we modify the T cells, so what we introduce in these T cells. So in the first example, the patient, there is a patient with a tumor, and the cells from this patient are isolated, and the TCRs that recognize, for example, a tumor antigen are then cloned and using a viral vector in this example are transferred to other T cells. And so the specificity of this TCR is then transferred to a new cell. And this new cell can then be expanded and give it back either to the same patient or to a different patient that have a similar cancer type with the same antigen. Another approach is to use antibody or like chimeric antigen receptor, where you have in this case an antibody that recognizes an antigen. A very famous one is the CD19 CAR that is now an approved therapy for leukemia and lymphoma. Then another possibility is to raise this T-cell receptor in animal models that express molecular or like MHC molecules that are human. And you can vaccinate this animal with a certain antigen, like tumor antigens or viral antigens, and then isolate the T-cell receptor that are specific for this antigen, and then transfer to a human cell and then to a patient. So there are a few important aspects to consider for this type of therapies. And I just listed what I think are the most relevant, but there could be still others. So in particular, we think that targeting the proper antigen is very important. And I will go more in detail about this. Then also another important aspect is how you perform the genetic transfer. So you can use viral vectors, non-viral methods, and how you do this may influence the efficacy of your therapy. And then effector function in vitro that we can measure by killing assay or other type of assay often do not correlate with the in vivo efficacy. So do we need some kind of model to predict this treatment efficacy? Can we improve the immunotherapy with modified T cells if we have a model before going into the patient that can help us identify the best candidate cell product, for example? And then another aspect that is very often neglected is the feasibility of the process. So this is that the problem is that there are several researchers that have great research protocol that function very well when is in the normal research setting. But if they try to get that protocol or that type of reagents into a GMP setting, so a good manufacturing practice or like the clinical settings, then the protocol sometimes or actually very often needs adjustment. So simple things like changing the reagent to make them injectable into patient, or for example, if the process requires a lot of steps, it may be difficult to actually use the same process into GMP condition when the people working, like the staff working in the GMP condition, they need to have like a space suit and it's very cumbersome. So when one thinks about modified T cells, also how you make this cell product is important, because one needs to think how you can arrive to, for example, a phase 1 clinical trial, but also how do you move after that. So increasing the number of patients that can benefit from this new therapy. So the types of antigens that have been targeted with immunotherapy are like mainly 3 types. So tumor-associated antigens. These are antigens that are overexpressed on cancer cells, but they are also present in normal cells. And targeting these antigens could be risky for the patient unless they are expressed on non-vital tissue. The example is CD19 that is also expressed on normal B cells. Then there are neoantigens. These should only be expressed on cancer cells because they come from the mutation that actually made the cell a cancer cell. So they should not be expressed by normal cells, and so there should not be risk for on-target toxicity. There could still be a risk of off-target toxicity due to cross-reactivity of the T cells. Then another category is viral antigens. These are only expressed by infected cells, but it is not known if they are also always expressed by virally associated cancer cells. So when one look at hepatocellular carcinoma, and this can be done just looking at the COSMIC database, the most frequent gene, or like the most expressed gene, are actually kind of known oncogene or driver genes for cancerogenesis, like TP53 and beta-catenin. And they are also quite often mutated in these patients. So in what concerns like TP53, for example, it is well known that several cancer types have mutation in p53, but what was still not clearly established is that how this mutation then results into antigens that can be recognized by T cells. So are these mutations immunogenic? And there are like recent papers now, recent studies that have been conducted in the surgery branch in Dr. Steven Rosenberg's laboratory that highlight the immunogenicity of mutation in p53 in different type of cancer patients. So like ovarian cancer, colon cancer, and other epithelial cancer. And in particular, I want to describe more in detail this last study, because in this study, for the first time, we see a systematic screening of the mutation occurring in p53. So the first question of the study was, in the patients that we see in the clinic, in this case in the surgery branch, how many of these patients have mutated p53 and what kind of mutation are in there? And so about 65% of the patients evaluated have mutation in TP53, and about 71% also have missense mutation, so that they change the amino acid sequence of the protein. So they could be seen as a foreign antigen and maybe become immunogenic. And so the approach that was taken was to make synthetic genes or peptides that contain the sequence of this mutation, and then evaluate the T cells of the patient against these artificial antigens. And both synthetic genes and also peptides were provided by GeneScript in this type of study. And it was interesting to see that among all the TIL screen, about 39% had some reactivity against the p53 mutation that could be measured by either interferon gamma secretion from the T cells or upregulation of activatory molecules like 4-1BB, like in this example. And then the next question was, but if this reactivity was found with this kind of artificial screening, because it was anyway a peptide-loaded antigen-presenting cells or a transfected antigen-presenting cells, so could these T cells recognize a autologous target, for example, a tumor cells from this patient? And the answer in this case, in this example, was yes. So it was possible to also demonstrate that these T cells could recognize the tumor, autologous tumor from the patient. So the in vitro assay for the efficacy of this T cell product showed actually a promising result. Now what needs to be tested is the efficacy in vivo. So what happens if you now introduce this cell into the patient? But the point of this study was also to create a TCR library, because if you screen several mutations, several patients with several or different HLA restriction, now you can start building a library of T-cell receptor that match a certain mutation with a certain HLA restriction. And so you can think about using these TCRs not only for the patient where you find them, but also as off-the-shelf reagents for other patients that have the same mutations and the same also HLA restriction. So then there is another step in the process toward a successful immunotherapy. And this is how you transfer then the T-cell receptor, or like your library of TCRs, into new recipient cells. So there are several methods. The most common one are retroviral vectors or lentiviral vectors, but you can also use DNA electroporation with the Sleeping Beauty plasmid system, for example, or RNA electroporation. And this last one is only a transient expression. And in the case report, I want to show you below here. This is like an example of immunotherapy for hepatocellular carcinoma, where a patient was treated with T cells that express a TCR, and this TCR was against hepatitis B antigen, and the TCR was introduced by RNA electroporation, so it was a transient expression. And this was done also for a safety reason, because this was a very pioneer treatment for this type of patient. So they also didn't want to have these T cells with the expressed T cell receptor too much, let's say, in this patient without any other safety reason. In this case, they knew that the cells would stop expressing the TCRs within a couple of days. So this patient was diagnosed with hepatocellular carcinoma, and he was also chronically infected with hepatitis B. He received a liver transplant with an HLA mismatch liver, so the donor and the recipient didn't have the same HLA. And then after about 10 years, this patient started to have distant metastases that were HCC metastases, still positive for HBV antigens, and they belong to the HLA of the recipient, so not the donor. And so the therapy that was done was to transfer these modified T cells that express the TCR against HBV, and it was possible to measure some transaminase increase in the patient, and this is a measure that indicates some liver damage or hepatoma, also like hepatocellular carcinoma damage. Then at the same time, they could observe a decrease in the viral antigens titers, and they could track also this T cell. The problem here is that the patient, unfortunately, didn't have any clinical benefit from this therapy. And so this brings to the next question, like, can we predict if a product, a cell product, will have any efficacy in vivo? Can we use any model before infusion to see if we are going to be successful? So here I just put as an example a very simple model that we use for hepatitis C specific T cell receptor. So this model is an hepatoma, human hepatoma cell line that expressed the HLA molecule, human HLA molecule, and then the hepatitis C replicon coupled with luciferase. So when the replication is active, these cells are actually luminescent. And we did the co-culture, so we isolated T cell receptor against this antigen, NS3 and NS5A. And we did the co-culture with the TCRs that we isolated and this hepatoma cell line. And for in this case, like 4 TCRs, 2 against each antigen, we could observe a decrease in luciferase. So both of these groups of TCRs were able to stop the replication in this model. Then we also measure the transaminase in the supernatant as an indication of toxicity or killing of this particular cell type. And we saw that only the TCRs targeting NS3 antigen were able to actually kill these cells. So this simple assay told us that there was some difference in these TCRs. And this could help in, for example, deciding in what kind of therapy you could use these TCRs. So, for example, the NS3 specific TCRs may be used for cancer therapy because they are able to kill cancer cells, but the NS5 TCRs can only maybe be used for fighting chronic infection, where you don't want to kill the cell or the hepatocyte, but just stop the viral replication. And this is only a speculation. We never really tested in patient these TCRs, but this is a way to evaluate different candidates for cell therapy. And then another model that is very interesting is a 3D microfluidic model. And this was designed by one of our collaborators in Singapore, Andrea Pavesi. And this device that he designed is a small device that we can simplify as a device with two different areas, like an inner areas where you can have like a matrix, a gel, where you can see, for example, cancer cells. And this matrix can be changed depending on the cell type that you would like to test. And then there is another area where you can inject the T cells that you want to use for your therapy, for example. And then you can observe if the T cells can spontaneously migrate towards the target or if they don't do it. And another way of using this device is to add now different conditions that can mimic the tumor microenvironment better than just a Petri dish. So, for example, you can add other cytokines. And in the case of liver cancer, we could be interested in adding these inhibitory cytokines and see how that affects our modified T cell product. And then it's also possible to test a combination of therapy. So, for example, modified T cells together with checkpoint inhibitors and so on. And what now is being evaluated is that using this model can predict the efficacy of the T cells in vivo in the patient. And then the last aspect that I would like to talk about is process feasibility. And this part is taken quite seriously here at Karolinska Institute. And that's why they designed this pre-GMP concept. And they opened recently the pre-GMP facility, and where I was appointed as director. So the purpose of this facility is really to support the research project that aim to go for clinical trials. So all sorts of procedures that are developed in the lab, but then they aim at becoming a potential therapy for the patient. So what we try to do and is to really offer as much support as possible for these researchers, so they can easily and safely translate their protocols to the clinic and start a GMP production and also eventually a clinical trial. And we have 5 fully equipped rooms that can be used for cell expansion, so if one thinks about cell therapy, but also vector and plasmid production and purification. And we have, as I said, like 5 different rooms, so we can carry on multiple projects at the same time. And other important aspect is also to help not only the process itself, but also all the documentation that goes together with being, for example, approved for a clinical trial. Other important aspects are test new equipment, for example, and also educate and train the staff that will have to also do the GMP production. So now I will describe more in detail the strategy that we have here for hepatocellular carcinoma. And we can start with the sample, or where we get the hepatocellular carcinoma sample. In this example is from a liver transplant. So the tumor cells are then isolated and used to sequence the mutations present in relevant genes, so TP53, beta-catenin, TERT, and others. Then the mutation that we identified with the sequencing can be used to make synthetic peptides. And again, the services provided by GenScript are very useful for us for this type of screening approach. And once we have these peptides, we can load them into antigen-presenting cells and then perform an immunological screening with cells coming from either the same tumor sample, so tumor-infiltrating lymphocytes, but also lymph nodes, for example, or blood of the patient. So again, when the T cells show reactivity against the antigen-presenting cells loaded with peptide, we then isolate the T cell receptor. We aim at collecting a library of T cell receptors that recognize different mutations and are also restricted by different HLA molecules, so that we can offer a broader like set of T cell receptor for different patients with different mutation and different HLA restriction. And then in the pre-GMP context, we can start to develop our process and try different things. So try, for example, different ways to introduce the gene into the lymphocytes. We can try to use different lymphocyte populations, so not only T cells, but also mate cells or NK cells. And then we can evaluate the feasibility of this process before going into real GMP production, then try a clinical trial for advanced HCC. So now a few concluding remarks. So promising preliminary results from current HCC immunotherapy indicate that it is possible to unleash the immune response to fight cancer. It is unknown whether there is an immunological response against neoantigen in HCC and if it correlates with clinical benefit. Our hypothesis is that the potentially relevant target antigens for immunotherapy are mutations in driver oncogenes. And we also think that a combination therapy of different immunotherapies, for example, modified T cells together with vaccines or checkpoint inhibitors, would probably be the best approach for HCC and other cancer types. And with this, I would like to acknowledge my former colleagues at the National Institute of Health and the very talented scientist in Dr. Steven Rosenberg laboratory, and also other former surgery branch members that now are independent researchers. Then our collaborators in Singapore and in Italy, and my current colleague now at Karolinska Institute and Karolinska University Hospital. And thank you also, Tracy and the GenScript for this opportunity to give this webinar. Thank you.
Thank you, Anna, for the fascinating research. This is Dr. Minnie Hu. I am the product manager of GenScript's peptide service. So before we get into the Q&A session, let me take this opportunity to introduce GenScript's capability for peptide synthesis. GenScript offers comprehensive peptide synthesis services. With our 15 plus years of experience, we have delivered 600,000 more peptide delivered to over 10,000 scientists worldwide. We have also collaborated with over 3,000 peers publications. So we offer customized peptide synthesis, including regular peptide synthesis and upgrade to express peptide to be delivered in as fast as 5 days. We also offer peptide library synthesis, including the standard peptide library and also microscale peptide library. So now with our peptide PepPower technology, we have guaranteed over 98% synthesis success rate and also a sequence length up to 200 amino acids. And we have 100% guaranteed quantity to be delivered. Okay, so we start seeing some questions coming in for Anna's talk. The first question that we have is, Anna, how do you exactly perform a functional immunological assay?
Okay, so it's quite complicated, but we can simplify by saying that is a co-culture of target cells that can be antigen-presenting cells loaded with peptides or transfected with an antigen that you want to see if it's recognized. And then you co-culture these target cells together with what you think is like your effector cells. So it can be T cells coming from the tumor in itself of the patient, coming from the blood, or your modified, for example, T cells. And then if the T cell recognize the antigen on the antigen-presenting cells, on the target cells, then it gets activated, and you can measure this activation by either checking a cytokine secretion like interferon gamma, so you can do an ELISpot or an ELISA, for example, or you can also measure activation molecules on the surface of T cells like 4-1BB or OX40 or CD107, and this you can just stain with antibodies and then measure by flow. This is the process.
Thank you. So another question is, what if our audience want to use your pre-GMP facility?
Okay, so the pre-GMP facility is located in Sweden, in Stockholm, at Karolinska Institute, and it's an open facility, so one can really, first of all, I would say contact me. So my email is anna.pacito@ki.se, and then we can arrange a meeting and then see what is the need for these researchers. So like if they need more advice or support on the protocol itself, finding reagents that are GMP compliant, or if they want to use instruments that we have in the facility. We are equipping constantly the facility with the latest instrument available for cell expansion, cell growth, and detection of particles, so a lot of different possibilities. And then basically we can discuss together how to better proceed and give the users that is interested the best support ever at every step of the process they may need.
That's great, and we will also put Anna's contact information on the transcript. So due to the time consideration, we will follow up on other additional questions. And thank you again, Anna, for the talk. Very nice. And thank you for all of your attending of our webinar. And we hope you find this webinar useful for your research. And again, we would love to hear your feedback. So please help complete the survey after the webinar